In this video, we will discuss about the Quiescent CD8 plus T cells and Effector CD8 plus T cells. What are the difference in these two CD8 plus T cells conditions? And here we will discuss a review paper that has been published, which discusses about two independent studies, or which summarizes the results of the two independent studies. So here, what they have explained in this figure is that this Quiescent CD8 plus T cells, or the CD8 plus T cells, which has this stem cell like behaviors, that is, they can self-renew and replicate or proliferate themselves. So the reason, one of the features of this Quiescent CD8 plus stem cell like T cells is that they have this high level of this SATB1, and this high level of SATB1 will make sure, or will help this CD8 plus T cells to remain in its stem cell like quiescent state. And the markers for this condition is this CD62L ligand and this TCF1 positive. So CD62L positive and TCF1 positive, these are the markers for this stem cell like quiescent CD8 plus T cells. Stem cell like quiescent CD8 plus T cells here, right? So once they differentiate into the effector cells, in that case the cell surface marker changes to this CX3CR1 positive. In that case, they are the effector T cells, or effector like CD8 plus T cells, and in that case it can act against the tumors, or it will have a better anti-tumor efficacies. And in these conditions, the amount of this SATB1 is reduces. So what actually happens is that once the differentiation of this stem cell like quiescent CD8 plus T cells occurs into the effector T cells, the amount of SATB1 levels decreases. So this is the feature of this SATB1, that once it is decreases, then it will have the less then the commuted lineage CD8 plus T cells will have the less self-renew and proliferation ability. In some cases, the differentiation also occurs to this terminal CD8 plus T cells that has the CD101 markers, and the condition in which this CD101 terminal T CD8 plus T cells forms or differentiate is when the patient is at the advanced stage of the disease, or the advanced stage of the tumor development. In that case, the CD8 plus T cells is differentiate into this CD101, and this kind of terminal CD8 plus T cells has the less ability to kill the tumor cells, and they are less responsive towards the immunotherapies such as this checkpoint inhibitors immunotherapies. So in summary, what they are the point that the review paper is making, that the stem cell like CD8 plus T cells has this high level of SATB1. However, the differentiated or effector T cells has the low SATB1 level. So one can use this informations to suppress these SATB1 levels in these stem cell like quiescent cells to have the more of these effector T cells that can act against the tumors and increase the anti-tumor efficacies. And another way to think about this is that high SAT1 levels will reduce the anti-tumor efficacies, or it curbs down the anti-tumor efficacies of the CD8 plus T cells. So in order to increase the anti-tumor efficacies, one can target this SATB1 level in the CD8 plus stem cell like quiescent CD8 plus T cells. So that way they will have more effector like T cells. So however, the detailed studies needs to be done, because the long-term effect of mutations of this stem cell like CD8 plus T cells, it still needs to be investigated before it can go for a commercial trials. So I hope you understood the difference between quiescent CD8 plus T cells and effector CD8 plus T cells. If you need to understand more about this topic in detail, you can go through the reference. that has been provided in the description. That's all for this video. Thank you for your time.